You know, you may be right about the house tilting. That is getting us into scientific method. Is that the actual nuts and bolts of how to set up and conduct and interpret and do all that sort of stuff with science. Observation. There's three categories of observation. One of them is direct observation, which means that my direct observation is coming into my brain through my actual personal senses. So it's something I see, or something I hear, or something I touch, something I taste, that's direct. It's going directly into my brain. Indirect, coming into my brain secondhand. It could be someone else's observation. They tell me about something, or I read about something. I look at somebody else's results from their experiment, and that becomes my observation. That's one type of observation, where it's coming in from other people one way or another. The second type of indirect observation, we talked about this. Spontaneous generation was easy to believe because there were a lot of things too small to be seen in the world by human eyes. But we have microscopes now. We have all sorts of technology that allows us to observe the world in a way that our senses can't. That's indirect. It's coming in secondhand through our devices that we have to trust are working properly, that are giving us accurate information. So, indirect is always a little unreliable. Direct sometimes is unreliable. You can't always trust your own senses, or at least the way that you make sense of your own senses. But that's your starting point. You make some sort of observation about the world, and then you go with a hypothesis based on that observation. Leads to a hypothesis. Here's an observation I made in this particular summer. Some of you, I've talked to other people, and apparently it's not just me. There are a tremendous amount of rabbits and a decent number of other small furry creatures. And a lot of times small. I've never seen so many baby rabbits running around. That is an observation and a bit of a judgment. Is that I'm comparing to my memory, and I'm pretty sure I'm right that I typically haven't seen this many little bunnies running around. But that's an observation. What does that mean? Well, I do have a hypothesis. I also have not noticed a lot of hawks. And that may be why these bunnies are getting out as babies and getting big enough for me to notice them, because they're not being picked off by hawks, which are major players in the anti-bunny crusade. True for a lot of the other small animals as well. I've talked to bird people. They say there's nothing really going on with the hawks in general, but I have looked for hawks around here and not found them. That's ties into the two things your hypothesis should have. So my hypothesis is that the rise in the bunny population is somehow related to the drop in the hawk population. Which means if I started looking systematically all over the place for hawks, that I shouldn't be able to see very many of them. That if I spread my net a little bit bigger and talk to people, like, go out and look for hawks. Is it, tell me what you can find. Is it, do you normally see them in the summertime? Are you seeing them in the same place? Is that, and my idea from my hypothesis is they're not gonna find as many of them. But what I'm doing using that is I'm developing a test. Now, the type of test I just suggested is the type of test the biologists and a lot of sciences wind up having to run. Is that there's the classic controlled experiment that you can do when you've got, okay, here's where we run into trouble. Is that there's a control that's a comparison test, and there's the control which is your ability to control what's happening. And unfortunately, they wind up getting smushed together, and it's hard to puzzle them apart. So even I'm gonna use the words in ways that I probably shouldn't. I should come up with, trying to come up with a better word for our ability to see, I can't come up with anything other than to control the situation that we're doing. The test that I suggested is a field test. It's only testable out in the real world, and so there's a lot of design stuff I can't really do with it. I could, if I had the money, hire a bunch of expert observers to go and run surveys on the hawks. My problem is that I then would be comparing it to just loose observations that people had in previous years, so it wouldn't be a good comparison. It's actually better to take people who have volunteered because in their area they are bird watchers or they're good observers, and they have some rough idea, the same way that I do, of what your general local hawk population is in a typical summer. And then they can tell me if this year seems lower. It's not great, but it's the best we can do. And that's what happens with field tests, where you have something you want to test about the real world, but you can't bring the real world into your particular laboratory. It may be too expensive. It may be impossible to duplicate. It may not be ethical to bring wild samples of maybe something that's endangered into laboratory conditions. It may just be too doggone expensive. So a lot of biology is done with field tests, and that puts a lot of restrictions on how you can design a test. Because the classic scientific method test is an experiment. And I guess I should put a classic experiment, because a field test is still an experiment. It's just given that designation, you understand that there's issues involved. So we want to make a comparison. We want to run a test and then a comparison test to see if there are differences. Well, differences based on what? Differences based on some part of the hypothesis that you can bear down on, you can focus on. My hypothesis has to do with hawk populations. You know, I think it has an effect on the bunnies, but that's actually incidental, is that my hypothesis is that the hawk populations have dropped. And I could do a follow-up on that to see whether there's some reason why there's lowered hawk predation on bunnies. If it turns out that the hawk population is the same, now I gotta go back to the drawing board and try to explain my original observations of there's lots of bunnies around. So I already gave you a rough outline of my field test. What am I comparing? Hawk populations in the past to hawk populations today. That's what I'm going to be testing, and that's what I'm going to be, that's what's going to be changing, one thinks, between the group that's going out and counting them today and their idea of what it was like in previous years. Is that this is why field tests are fuzzy, is that that's not the best approach, but it's the best I could do under these conditions. More straight up, say you want to test hydroxychloroquine. The way that it should be done is you take a bunch of people who potentially can get exposed to coronavirus, and because it's supposed to be an early intervention treatment, and you split them up into a group that's going to get hydroxychloroquine and a group that's going to get something they think is hydroxychloroquine. We'll come back to that in a second. And then you see whether COVID plays out the same way in both groups, deciding on how do we decide whether COVID has played out the same way. Do you go on positive tests? Do you go on hospitalizations? Do you go on serious hospitalizations? It all depends on what you think the hydroxychloroquine is good for. Does it prevent the infection in the first place? Does it lower the progression of the disease in some significant way? And if it does, what are you going to measure to decide that? But what's the variable? Hydroxychloroquine in our test group, a fake hydroxychloroquine in our control group. And if something happens in the experimental group that doesn't happen in the control group, or it happens at different levels, then the question is that a big enough difference to mean anything? But the variable there would be the hydroxychloroquine. One of the early things that you may want to decide is, okay, maybe this is iffy to use on people, or it's a broad test that I can't really run in the real world, or it's supposed to, in order to run it right, I'd have to run it for years and years and years and years, and I'll never get the funding for it, and I won't live long enough to run it for that long. So can I use some sort of stand-in as a substitute for the test I really want to run? Stand in for people? Maybe we can run a test on mice. Mice are mammals, they're furry creatures like us. They have a lot of the same chemistry that we have. They don't exactly match us. That's the problem with animals. In the case of coronavirus, it turns out mice don't really get the coronavirus, but hamsters do. Hamsters turn out to be a decent animal model for coronavirus. Ferrets get a related coronavirus. Is that really a good comparison? This is another problem you wind up with, is that the animals don't get exactly our virus. They get a related virus that isn't exactly working the same way. In the case of HIV, that causes HIV/AIDS. Monkey and ape models, apes especially, get what's called SIV, simian immunodeficiency virus. Doesn't work exactly the same way, but sometimes it's the best thing we have as a comparison for tests we can't really ethically run on people. And sometimes we can take, if we're really trying to figure out what something is going to do, is use bunches of cells that we can get to stay alive in the lab and dose the cells directly and see whether something happens. Now, cell cultures are literally dishes or vats of cells, and so they're divorced from connection to a whole system. So they do behave somewhat differently, but it can give you some rough idea. It's not a good substitute. Animals are an okay substitute, but not a great substitute for people. Cell cultures are even worse. But for early drug discovery, it's kind of the best that you can do. Now, for big, expansive tests, oops, if I can spell, you may go to computer simulations. We want to know what's going to happen to the world climate if carbon dioxide levels, emissions continue at the current rate. Or what happens if we can get them under control, or what happens to the climate if the oceans do start to rise. What happens to the climate if the permafrost in the Arctic starts to thaw out, what's going to be released. And those are huge computer models. And they're limited in that you have to decide what the computer is going to be using, what sort of information. You have to tell the computer how things work, and sometimes you don't know how things work. Or there's things that are going to be important that 30 years from now you go, wow, that was important. We never knew that to put it into the computer models. So simulations are useful. They can, in some cases, simulate a human body and tell you, okay, the drug may do this, this, and this. But again, the limitations of our knowledge is the limitations of the computer's knowledge. It can combine and do lots of calculations that we can't do faster than we can do them. But it's still limited by what we do or don't know. But they're very, very useful as kind of early stages of tests before you can run tests or watch how things play out in the real world. Your control test or tests is your comparison test. Ultimately, you want to be able to change your variable between your real test and your control test. It'd be great if you could just take the variable away. You can do that with drug tests, right? Hydroxychloroquine and then non-hydroxychloroquine, but a duplicate where they think that they're getting the drug. We'll talk about confounding factors in the next video. But your control test is your comparison. Because if we run a bunch of people with a certain drug and we see stuff happens, we can't be absolutely sure that the drug is doing what we think we see happening. But if we have a group that's not getting the drug, and then we compare it to the group that's getting the drug, now the differences we can pin to our variable. That's why it's called a variable. It varies between the tests. Some cases you may vary it a few different ways and then look for differences in the results in a few different ways. But your control test, like Reddy's 2 different dishes with meat in it, is a very, very valuable part of scientific method. And we'll pick up the tricks of this and the issues in the next video.